topic. Sometimes you can expect some questions also based on this topic, short type questions as well as essay type question. So as per a sequence alignment, from yesterday onwards, from the last few classes we were doing with the sequence and NCBI GenBank and lot of sequence matching also we have done. Yesterday you have seen how a phylogenetic tree can be obtained with the help of DNA sequence. So we have to first understand what is the meaning of the word sequence alignment. A sequence alignment is a way of arranging the sequence of DNA or RNA or a protein to identify regions of similarity that may be consequence of functional, structural or evolutionary relationship. Here you can see that there are two sequences here. We have written that, and if you write that sequence in a particular way, we can find that certain words are similar in both sequences. This way of arranging the sequence one above the other to find out the regions of similarity is known by the name sequence alignment. So here two English words are kept one above the other. So we can see that some kind of sequence similarity is there. And purpose of that sequence similarity is mainly because of the structural similarity between the organism. Or there can be the evolutionary similarity. For example, if you take a cat and you take a lion also, both are coming under the cat family. You take a dog and a wolf, both are coming under the wolf family or dog family. We can find a lot of similarity. But if you compare a cat with a dog, we cannot find so much similarity than we can find between two cat family members or a wolf dog family members. So even if there is some evolutionary similarity, that similarity will be reflected in the sequence alignment or in the sequence similarity. Aligned sequence of nucleotide or amino acid residues are typically represented as rows within a matrix that we will know. Then we are also putting gaps where there are no sequence similarity. That point also we will discuss later. So sequence alignment, how you will be defining a sequence alignment? It is a scheme of writing one sequence on top of another. Here we are writing these two English words and we are writing it one above the other, but residue in one position are deemed to have a common origin. See, if you are writing it like this, in the case of B, we are not able to find any sequence. So what we are doing is that we are putting a gap there or a dash there. So that gap indicates that there is no similarity. There is absolutely zero similarity there. Where the words are written properly, we can see that A is matching with A, N is matching with N, A is matching with A. So that way we can find that there is some similarity between these different sequences. That way of arranging or that way of writing the sequence one above the other is known by the name sequence alignment. So here banana and ananas, two English letters, are placed one above the other, and wherever we are finding this similarity, this area indicates that there is a lot of similarity. That means that area shows evolutionary similarity. That means which are the traits or characters represented by these genes will be similar in both species. That is the meaning of this alignment. Now we will be seeing one more. See, if you look at this, then will you be able to read that it is an English sentence. If you try a little bit closer, you will be able to find out what is the meaning of that. It is an English sentence. Yeah. Some people easily got it because you know that sentence, so that you are trying to find a meaning in that. So here it is not written like truth is more important than fact, but by your previous knowledge, by your intelligence, you are trying to find a meaning in that. So this way also scientists are trying to find a meaning in the evolutionary sequence. Only when we are reading it in a particular way, the sentence becomes meaningful. Otherwise, there is no meaning in that. So here, the truth is more important than fact is a sentence that you already know. So you are trying to, so when you see that you are putting gaps wherever it is needed, it is not shown here. It is your mental calculation or mental breaking up. You are breaking up according to the way your knowledge, according to the way your mind works. So that is why you are finding a meaning in that. So only if we are putting the evolutionary sequence or DNA sequence in a certain way, there will be some meaning in that. That is why we have shown this. Here we can find different sequences are there. Two ways the sequences are arranged here. This is also a different way of arranging sequence. Yesterday we have seen this kind of sequence, and this kind of arrangement is known by the name multiple sequence alignment. Here more than one sequence is aligned with other sequence. So if two or more sequences are aligned this way, we call it by the name multiple sequence alignment. Here there are different types of sequence alignment. The first one indicates the pairwise sequence alignment. What is the difference between the two? Here only one pair is aligned with another. Only one sequence is aligned with another sequence. So a pair of sequences are taken into consideration. Here more than two sequences are combined, or more than two sequences are aligned with one another. Then we call it by the name multiple sequence alignment. Then we can again see that there are two different type of alignment. One is a feasible alignment. Feasible means possible. Other is an infeasible alignment, means less possible, not practically possible. That is the meaning of the. So here you can see that this is the way the sequence alignment is placed at A, T, C, C. Then the gap is inserted here. Then G, A, G, E. So this is the most possible type of alignment that is practically possible in nature. That is why you are putting it as a feasible alignment. On the other hand, if we go for this kind of alignment, we have to put gap here, three gaps we have to put. So an alignment that reduces the number of gaps are always practically more possible. So that is why the first alignment is said to be feasible alignment or more a possible alignment, because less number of gaps are there. In the second alignment, more gaps are there. That is why it is considered as a less possible or infeasible alignment. Same kind of appearance is also here. You can say that only fewer gaps are here. Whereas here, if you look at that, there are a lot of gaps are there. So as the gaps are increasing, the possibility decreases or feasibility decreases. That is the meaning of this feasible alignment and unfeasible. So evolutionary speaking, an alignment that shows only minimum number of gaps is always practically or evolutionarily possible. So pairwise alignment means the Corey sequence. What is a Corey sequence? You can consider this one as a sample sequence is aligned with a homolog sequence from a database. Here only two sequences are aligned. So that is why this is a sample sequence or the Corey sequence. It is only aligned with another sequence. Then multiple sequence alignment means more than two sequences are aligned with the Corey sequence for getting a Corey set. So that is how. So more than two sequence. Here you can see that this is the left side, right side is indicating multiple sequence alignment. Left side is indicating the pairwise. Only two sequences are indicated. But if more than two sequences are aligned with one another, we are calling it as a multiple sequence alignment. Then, so what is the meaning? What is the purpose? Or the pairwise sequence alignment are used to find the best matching local or global alignment of two Corey sequences. We have to understand what is this global alignment and what is this local alignment. Here the different type of pairwise alignments are shown here, and here you can see that this is considered as a global alignment and this is considered as a local alignment. The only difference is that in case of global alignment, an attempt to align every residue in every. Here you can see that this is the Corey sequence and this is the next sequence, homologous sequence. What we are doing is we are starting from the one end and stretching it up to the opposite end. So here completely stretching it. So that way, if we are aligning every sequence in this way, we call it by the name global sequence alignment. But what we are doing in the second one, We are only aligning where there are maximum similarity. This same sequence can be written in a different way. This is the first one where the entire sequence is aligned. That is why global alignment. Have an attempt to align every residue in every sequence. So whereas in the case of local alignment, parts which are showing greatest similarity are only aligned. In other regions we are putting the gap. So here you can say in this region there are more similarity, and here there is no similarity, so we put a gap. Here also some similarity, here there are no similarity. So regions which are showing more similarity are always aligned, and other regions are left as gaps. So that kind of alignment, where alignment is done, where there is greatest similarity, is known by the name local alignment. So in the case of pairwise alignment, there are two types of possibility. One is aligning the entire region, which is known by the name global alignment, or aligning only the region where which are showing the maximum similarity, that is known by the name local alignment. Then optimum alignment. Optimum is something that we may have studied from chemistry class. Optimum temperature and all. So optimum means the most ideal, is the best alignment according to defined set of rules, and there are number of rules. For example, number of gaps. Less gaps are always ideal. More gaps are not less favorable. So that is why an alignment that favor less gaps are always aligned. So that way we are finding out which is the best alignment, or which is the most useful, most possible alignment. So it is ideal for comparing different sequences, and several kind of scoring matrices are there. And that area we are going, how this optimum alignment is discovered. What are the parameters, or what are the techniques by which we will be able to find which is the best alignment, or which is the best optimal alignment. So multiple sequence alignment is basically an extension of pairwise alignment, and the only difference is that more than two sequences is there. So so that is a basic difference between pairwise alignment and multiple. Here you can see that this is a diagram showing multiple sequence alignment of several species. You can see that some regions are showing maximum similarity. In some regions there are no similarity, so that we are putting gap. So this way, yesterday also we have seen how multiple sequence alignment was possible. You can go to the NCBI and you can be using Mega software. You can easily find out your own multiple sequence alignment from some 5 or 6 sequence you are taking from GenBank. So that way it is possible. So sequence alignment is a way of writing the sequence of DNA or RNA to identify regions. So the ultimate purpose of any sequence alignment is to find the regions of similarity. So yesterday you are doing 10 species and you are trying to find the regions of similarity. What is the purpose? You want to show how similar these two sequences, these 10 different spider species are. That is your aim. And for that you are going to draw a diagram called phylogenetic tree, and for an expert, by simply looking at the phylogenetic tree, he will be able to guess how far this species 1 is away, is similar to species 2, or how different species 1 and species 4 are. Such kind of information can be easily obtained from a phylogenetic tree. So that is the advantage of a multiple sequence alignment. So in order to draw a phylogenetic tree or a phylogenetic graph, we need to have first thing, we need to produce a multiple sequence alignment. Based on this multiple sequence alignment, we are finally finding out finding out the phylogenetic tree or evolutionary tree that is possible with that number of species. So the lot of information can be obtained from a phylogenetic tree. The most important information is that evolutionary relationship. So if you take 10 species belonging to the different cat family, cat species, you can find a lot of similarity. You will be able to find out which two species of cat are more similar, which are more distant from one another. So that way you will be able to discover the evolutionary similarity or evolutionary relationship between different species. So that is the aim of any multiple sequence alignment. Sometimes it is abbreviated as MSA. So if you heard the word MSA, you have to understand that MSA simply means multiple sequence alignment. The primary purpose of MSA is in the phylogenetic analysis or in the preparation of evolutionary tree. Now what we are going to see is how we are doing a multiple sequence alignment for an evolutionary tree. So that is, earlier I have discussed that there is a term called optimum alignment. So there are different possibilities here. Out of these different possibilities, you have to find out which is the best alignment or which is the optimum alignment. For that, we are doing certain mathematical calculation. That mathematical calculation is known by the name scoring scheme. And to go into the scoring scheme, we are putting certain rules. The rule 1 is that if there are two match, for example, thymine and thymine are coming, it is a match. Cytosine and cytosine are coming, it is a match. Cytosine and guanine coming. So if there is a match, we will give a point of 2. If there is no match, we will give a point of 0. And if there is no possibility for a match or mismatch, we will be putting a negative value, that is -1. So this is the first set of rules on which we are going to calculate. So I just keep these rules in your mind. Now we are going to implement this rule in this sequence. So you can see that there are 3 alignments are possible. So this is the first alignment, this is second alignment, and this is third alignment here. 3 alignments are possible. So what we are doing is that where there is no match, we are putting 0. When there is no mismatch, there is no match or mismatch, we are putting -1. What is -1? Gap penalty. So gap penalty is the -1. Here also there is no gap, so we are putting -1 again. Here also -1 because there is a gap, and here there is no. Here this I and I are similar to each other. This I and this fifth letter I and I here are similar. So when there is similarity is coming, we are putting what we call it 2. When there is similarity is coming or match is coming, we are putting 2 as a letter there. So I indicates 2, N indicates also 2. So when there is similarity, we are getting 2. When there is no similarity, we are putting 0. When there is nothing to compare, we put it as a -1. So by this way, if you are calculating the arithmetic sum of all these numbers, we are getting a value called -3. So that is how this is how we are simply adding it together, summing it together. Then the second sequence also we are writing it that, what is the difference? This is we are writing it this way. This is another possibility. This is a longer sentence and this is a shorter sentence. We are finding certain regions only similar. That regions we are writing. Then we are finding that if you are writing these 2 sequences in this way, the value is changing. The value is improving to -1. So then here also you can find another possibility. This way, third value, the value we are getting is 3. So here we have 3 values. So from these 3 values, which will be the best alignment? Which alignment will be the best? One is -3, -3. Other is -1. Other is -3. Which will in your simple guess, what will be the which alignment will be the best alignment out of these 3 alignment? Can anybody guess? No, even the wildest guess. Just like you are answering multiple choice question, just say which is the best. First, second, or third? Which one? Which one? Say louder, Anjali. -1. What is the reason for that? -1 is a more positive value. That is the only reason. So -1 is more positive, -3 is more negative. So that is the reason why minus. So in alignment showing more positive value will be always taken as the optimum alignment. So this is the way by which we are finding out the best alignment. Now this is a very simple way of calculating. Now we are trying to improve that by adding 2 more rules to that. So what we are doing is that when there is a match, we will be putting the figure 2. And for the mismatch, we are giving, earlier what we were digging is that mismatch is always indicated as 0. Now we are going to redefine it in a more precise way. We are finding 2 types of mismatch. One is similar mismatch and there is dissimilar mismatch. If there is similar mismatch, we give 1. If there is dissimilar mismatch, we 0. The only thing that we are changing is that for mismatch we are giving different values, 1 and 0. For gap penalty it will be -1 itself. So based on the second rule, the modified rule, we are going to find out how this the same thing will be working again. So here we can find that for the when there is similarity, D and D are almost pronounced the same, DT. So that is why we are putting 1 here. So what is the rule? When there is similar mismatch, it is +1. When there is dissimilar mismatch, we are putting. So that is why here D and T, they sound similar. We are using English alphabets. So when it is similar, sound similar, we are putting 1. When there is a gap, we are putting -1. When there is here, for example, the C and Y, there is no similarity between the pronunciation of C and Y. So we are giving the value 0. So that is the difference between the second scoring scheme. So by trying the second scoring scheme, we are getting that for the first alignment we are getting -4. Second one we are getting -1. For the third we are getting -2. So here also we are finding that the second alignment, which are showing the -1, may be the best alignment. So in the first scoring scheme also we found the second alignment as the best alignment. So going to the second rule, implementing the second rule, we are finding that the second alignment will be the best alignment. Now we are again improving it again once again. Scoring system 3. So we are modifying it in a different way. So if there is a match, we always give the 0. And for mismatch, similar mismatch we are giving 1. Dissimilar mismatch we are giving 0. And for gap penalty, earlier for gap penalty we are giving -1 as the value. So here we are modifying that rule again. For gap penalty, we are finding 2 types of gap. One is a terminal gap, gaps which are located at the end. These are known as a terminal gap. This is located at one end. Gaps which are located in either in any end or terminal ends are known by the name terminal gaps. When terminal gaps are there, we give the value 0. When gaps are present in the middle, we are giving the value. So if this kind of gap, we are giving the value -1. So the only difference that we implemented is that we modified the gap penalty. For terminal gap we give 0. For middle gaps we are giving -1. So based on this third system, we are again modifying it, and we are seeing that by applying this again rule, we are getting more refined value. For the first step, we are getting a positive value of 2. For the second, we are getting a positive value of 3. For the third, we are getting a positive value of 2. So by implementing a third set of rules, we again found out that the second alignment can be the best alignment because it is giving higher positive value. So any alignment that gives a higher positive value will be always preferred than something that shows a less positive value. So this is an important rule in the use of this optimum alignment preparation. So this is how, and this kind of thing is done by the scoring matrices. Scoring matrices or scoring scheme is simply a mathematical way of calculating which is the best alignment or which is the most possible alignment. So we are again, then we are again modifying it in another way. The only difference is that opening gap we give the -3. For if the opening gap means if gaps are at the beginning, and if ending gaps means gaps are at the end, we are modifying it in a different. For opening gaps we are giving -3. For ending gaps we are giving -1. So that is the fourth set of rule. Going to the fourth set of rule, we are again modifying, and we are seeing that here the first alignment is getting -7, second alignment is getting -6, third is getting -9, and fourth is getting -10. Here also we see that second alignment has a higher positive value. So second alignment will be the best alignment under different circumstances. So this is the application or useful of scoring scheme. It is simply a mathematical way of finding or statistical way of saying that this is the best. So that is known by the name scoring scheme. So gap scoring is crucial. So nowadays we are not going to do it by our own. There are computer programs. If you simply put the sequence into the computer program, it will find out all these things. It will also show apply the scoring scheme, different scoring scheme, different rules will be applied, and show that this match will be the best alignment or most possible alignment. So that is the advantage of this this scoring scheme or scoring matrices. So then we have another is that there is a PAM is there, that area we will be discussing in our next class. So today we will stop with this multiple sequence alignment and also the scoring matrices. Sometimes you may, especially for your examination, you can get questions like distinguish between pairwise alignment and multiple sequence alignment, or give an idea of the different parameters to be considered in the preparation of a optimal alignment.